This is Dr. Fermani with the instructions on how to complete lab number 7 online. Lab number 7 is on DNA structure, replication, and protein synthesis. Lab number 7 corresponds with chapter 10 in your textbook. I highly recommend reading this chapter before beginning this lab and going over any lecture materials on chapter 10 before beginning this lab. This lab is involved and has a lot of information, so it's very important that you read the introduction and all the protocols before beginning working on this lab. Once you have done all those things, then you can begin lab number 7. Of course, like at the beginning of all labs, you have your pre-lab questions. So make sure, again, you're just really reading that introduction very carefully and you are able to answer all your pre-lab questions. So here you have pre-lab questions 1 through 9, and just like before, you'll come to your Word document. Make sure you tell me your name, that these are your pre-lab questions for lab number 7, and you can simply just type all your answers 1 through 9 for your pre-lab questions. Going on is now the beginning of the lab, the introduction that you have already read, and now we come to part 1, which is DNA structure and replication. So obviously, you're not going to be able to build the model at home. However, we can still go over the model and go over the parts of DNA structure. So in this model, it's a great 3D model to visually see the structure of DNA. Luckily, here is a picture, and we can still go over it together. So in your little kit, you would have these different pieces. So you would have all these different colored pieces, and each one would be a different base. So that is one component of a nucleotide. We know there's 4 different types. There's guanine, cytosine, thymine, and adenine. Okay, so you would have the 4 different colors. We know that guanine always base pairs with cytosine. That is called complementary base pairing. So G always goes with C and T always goes with A. Okay, so that is important to know about DNA. Here is the building block of DNA, also called a monomer. So this is called a nucleotide, and each nucleotide has 3 components that you have to know. So like we mentioned before, here's the base, a sugar, and a phosphate. Okay, so again, nucleotide is the monomer, the building block of DNA. We put lots of nucleotides together to get our 3D structure of DNA. In DNA, it's important to note that the sugar is called deoxyribose. Okay, so that is a specific sugar for DNA. So here's the completed 3D structure of DNA. There's a few important parts that I want to go over in the structure of DNA. As you can see, there are many nucleotides linked together, okay? And how do they bond in DNA? So again, like I said, the bases pair or bond with each other. You can see these small little pegs between them. Those are to represent hydrogen bonds. So as you can see, the nucleotide bases pair in the interior of DNA. We can see that here. So that is where the hydrogen bonds are. How do they connect on the outside is through the sugar phosphate backbone. You can see nicely that the sugar and phosphates bind nicely with each other, and that is through covalent bonds. Okay, that's in the sugar phosphate backbone on the outside of DNA. So those are very important to know, the chemical bonds in DNA, and also how do they connect to one another, okay? Through that sugar phosphate backbone, the sugar of one nucleotide bonds with the phosphate of the next nucleotide. Then that sugar bonds with the next phosphate, and so on, to make the sugar phosphate backbone with covalent bonds. Okay, so that's what I really wanted to point out for the structure of DNA. You can also see when it's built in this 3D position, you can see that it has that nice double helix. Okay, you can see that DNA is double-stranded. There's 2 strands, and that winds nicely into the helix form. Going on, you would use this model now. You would take it off that little pole and untwist the helix, okay? And now use it for DNA replication. DNA replication happens before before the cell divides in mitosis and meiosis. It only happens one time in both processes, okay? But it's very important that you make an extra copy of DNA so when the cell divides, it can each get a copy of DNA. So I can't stress enough that DNA replication is its own process, and again, it happens before cell division. So you would use this model to look at DNA replication. Essentially what you would do is see how our DNA is double-stranded. Here on the left is one strand, here on the right is another strand. The first thing that would happen is both strands would break apart, and there's a special enzyme that helps do that. It's called helicase. And remember, what are the bonds in the interior between the bases? Those are the hydrogen bonds. So helicase will come and break those hydrogen bonds to separate the DNA each into its own strand, okay? Now each strand can be used as a template strand. Okay, so here if we look forward, over here is the template strand on the left and the template strand on the right. It came from the original DNA molecule. There's another special enzyme called DNA polymerase that is going to come and read the DNA template strand. So in the template strand, if this was a base T, DNA polymerase would add a complementary A, okay? If this was G, it would add a complementary C, and so on. And now it starts to make this new strand on the left. This is called the daughter strand, okay? And it's the new strand and the old strand. That is why DNA replication is called semi-conservative, because again, this strand came from the original DNA molecule, and on the left is the new synthesized DNA strand, okay? And as you can see, as this process continues, the hydrogen bonds would be broken throughout this whole original DNA molecule, and what you're going to have is two DNA molecules. So we started with one and we ended with two. So that is DNA replication, literally making a copy of the original DNA molecule to now make two. There is a short YouTube video that I would like to show you on DNA replication. So we're going to watch this quickly. It's only a little over 3 minutes. It has some really good, a little bit extra information that is still really good to know, but really pay attention to how the DNA is replicated.
DNA is a molecule made up of two strands, twisted around each other in a double helix shape. Each strand is made up of a sequence of 4 chemical bases, represented by the letters A, C, G and T. The two strands are complementary. This means that wherever there's a T in one strand, there will be an A in the opposite strand, and wherever there's a C, there will be a G in the other strand. Each strand has a 5' end and a 3' end. The two strands run in opposite directions. This determines how each strand of DNA is replicated. The first step in DNA replication is to separate the two strands. This unzipping is done by an enzyme called helicase and results in the formation of a replication fork. The separated strands each provide a template for creating a new strand of DNA. An enzyme called primase starts the process. This enzyme makes a small piece of RNA called a primer. This marks the starting point for the construction of the new strand of DNA. An enzyme called DNA polymerase binds to the primer and will make the new strand of DNA. DNA polymerase can only add DNA bases in one direction, from the 5' end to the 3' end. One of the new strands of DNA, the leading strand, is made continuously, the DNA polymerase adding bases one by one in the 5' to 3' direction. The other strand, the lagging strand, cannot be made in this continuous way because it runs in the opposite direction. The DNA polymerase can therefore only make this strand in a series of small chunks called Okazaki fragments. Each fragment is started with an RNA primer. DNA polymerase then adds a short row of DNA bases in the 5' to 3' direction. The next primer is then added further down the lagging strand. Another Okazaki fragment is then made, and the process is repeated again. Once the new DNA has been made, the enzyme exonuclease removes all the RNA primers from both strands of DNA. Another DNA polymerase enzyme then fills in the gaps that are left behind with DNA. Finally, the enzyme DNA ligase seals up the fragments of DNA in both strands to form a continuous double strand. DNA replication is described as semi-conservative because each DNA molecule is made up of one old, conserved strand of DNA and one new one.
Okay, if we come back to your lab now, we can move on to part 2, which is on protein synthesis. Okay, so why do we even have DNA? So the whole purpose why we have DNA is because there's different sequences called genes, and each gene encodes information for the cell. In particular, genes code for proteins. So we're going to learn how the gene in DNA is going to code for a protein. Here is a diagram of the activity you would have done in lab. You can still understand this diagram and we can still use it so we can complete the work in this lab. So let's review this diagram. Okay, so here is our DNA template strand. Okay, so let me pause for a moment and make it very clear. Now I am talking about protein synthesis. This is completely different from DNA replication. The two processes do not happen together. They are separate. DNA replication happens before the cell divides. Protein synthesis happens all the time during the cell whenever the cell needs to make a protein, okay? So once we got that straight, let's go back here. And this is our DNA template strand, okay? So when we have DNA, it also has the complementary strand. So we know that DNA is double-stranded. Only one strand gets read to make protein. That is called the template strand, okay? So this strand will be read and will be used to make protein. So here, when we look at the template strand, it will be read by an enzyme called RNA polymerase. And as the enzyme is reading the strand, it will add complementary nucleotides to make mRNA. So again, RNA polymerase reads DNA to make it into a copy called mRNA. So mRNA is just a copy of the DNA. And as you can see, it's complementary. So in DNA, there's G. So in mRNA is a C. When there's an A in DNA, it binds to uracil in RNA. So as you can see, there's no T's or thymines in RNA. So again, A always goes with U, and then T goes with A, and so on, okay? So here's our mRNA, also called a transcript, and it's a copy of the DNA template strand. This mRNA is then used to code for amino acids. So how does that work? As you can see, the mRNA is chunked into 3 bases. These 3 bases are called codons. So codons are in mRNA, and every 3 bases codes for 1 amino acid. So how does it go from mRNA to amino acid? Is this kind of middleman called tRNA or transfer RNA. So tRNA will come and find the codon that corresponds to a particular amino acid. So this tRNA was GAU, so it will find this mRNA codon of CUA and bring along the corresponding amino acid. In this case, it would be leucine, okay? And again, so here is another codon, a different one. tRNA will come and bring the correct amino acid, arginine, and so on. And then this whole process, okay, of adding the amino acids reading the mRNA, this all happens in the ribosome, okay? So the ribosome will now link the amino acids together and you get a polypeptide chain. This polypeptide chain is not functional. It has to fold into a 3D protein to become functional. Okay, two words that I did not use that are so important are transcription and translation. So both of these processes happen one after the other to make a protein. Okay, so if I type out these words, I'm going to write transcription and translation. And I'm going to show you a trick to remember what occurs first. So transcription starts with trans and the next letter is C. Translation starts with trans and the next letter is L. So C comes before L in the alphabet. So we know that transcription happens first, then translation. So what are these words? So transcription is when that RNA polymerase is reading the DNA template strand and making it into a copy of mRNA or messenger RNA. That is transcription, simply going from DNA to RNA, okay? Translation is when the mRNA gets read by the ribosome to make amino acids. Okay, so you have to know those two words. You have to know they happen one after each other to make a protein. Okay, now that we understand this diagram, we can go on to a part of your lab to apply this diagram, to apply the knowledge we just learned, okay? So here in your data collection part 2, okay, we're gonna try to have a DNA template strand, figure out what the mRNA sequence would be and what the amino acid sequence would be. Okay, so for the first template strand, I am going to give you the information here, okay? And I also want you to do out the work in your Word document as well. As you can see, here were my pre-lab questions, and next I'm gonna do part 2, protein synthesis. Here's my template strand number 1, okay? Here is the sequence that I want you to focus on, okay? So you can read this, and now we're going to together make the mRNA sequence. I like to make my mRNA sequence in chunks of 3 in the codons, right? And it helps you, I'll show you in a minute, to make the amino acid sequence. So I'm going to read this template 3 bases at a time, and we're going to do this one together, okay? So the first one is CGT. So I know my first codon will be complementary. C goes with G, G goes with C, and T goes with A, okay? So we just did the first 3 bases. The next one is AAT, okay? Remember, A always binds with U in mRNA, okay? So A is U, A, U, and T goes with A, okay? So the next 3 are C, T, C. Complementary to that is G, A, G. Next we have ATA, that goes with UAU, and lastly we have GCT, which goes with CGA. So this is my mRNA sequence that I just chunked into my codons, my 3 base pair codons. Now, how do we read this mRNA to make the amino acids? And that has to do with something called the genetic code. So let's go to your lab manual, and on page 78 is this genetic code. Okay, you do not have to memorize it. It will be there for you to always go back to and use, but you have to know how to use the genetic code. So how do you use it is you always start from the middle and you work your way out. So let's go back to our document real quick. So our first codon was GCA. So let's practice. So here we always start with the first nucleotide, which was G, okay? So here, as you can see, it's this like light purple ring. My first base or nucleotide was G. Then I move on to the second ring. So the next one was C. So right after G, I want to connect it to the C, okay? I'm still staying within this quadrant that has to do with G. So the next one is C, okay? And as you can see, I'm only staying within this area that had C, and now I can pick one of the 4 nucleotides. So it was A, okay? So see how everything connects. G with C to A, and I start from the inside and go to the outside. So GCA codes for alanine, ALA, okay? So I come back and I know that my first codon codes for ALA, okay? That is my first amino acid. The next one is UUA. So let's go back to our genetic code. All right, my first base is U, so I start from the inside. Then I go to the next rank, so U, the next one is U, and the last one is A, okay? So UUA makes leucine, Leu. So I come back, okay, and I write leucine. That is my next amino acid. My next codon is GAG. Come back to the genetic code, GAG is glutamic acid, GLU, okay? So I come back and I write GLU for my next amino acid. Going on, UAU, let's see in our genetic code, UAU is tyrosine, okay? So I would write tyre right here, and then CGA, last, CGA is arginine, ARG, okay? So that is my last amino acid. So I made 1, 2, 3, 4, 5 amino acids, okay? These would all be linked together in the ribosome to make that polypeptide chain, and eventually they would be twisted and wound together to fold into a 3D functional protein. Okay, so we just did protein synthesis. We took DNA, we transcribed it into mRNA, and then we did translation to find the amino acid sequence for the protein. Okay, so going on, here's template strand number 2. This is the time that I want you to practice on your own, okay? So now in your document, please write out the template strand number 2. Here's the information right now. It's AAA, GGA, TAT, CAC, CCA. That is the DNA template strand. Now you're going to copy it through transcription into mRNA, and then you're going to take that mRNA and you're going to do translation to find the amino acid sequence, just like I showed you for the first one, okay? So please make sure you write this in your Word document. I'm going to be looking for it, okay? So that is for your own practice. Before we move on, I want to show one more quick YouTube video, okay? This is how protein is made in the cell from DNA. So let's just watch this as a recap to remember the process of transcription and translation.
Here is a cell, the basic unit of all living tissue. In most human cells, there is a structure called the nucleus. The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long strand of DNA, tightly packaged around proteins called histones. Within the DNA are sections called genes. These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of free bases in the nucleus. The DNA code determines the order in which the free bases are added to the messenger RNA. This process is called transcription. Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA. The messenger RNA then moves out of the nucleus into the cytoplasm. Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acid. Transfer RNA molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time. As each triplet is read, a transfer RNA delivers the corresponding amino acid. This is added to a growing chain of amino acids. Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein.
Okay, so let's go back to your lab and we can go on to the last part, which is on DNA mutations. So DNA mutations, like it says, mutations always occur in DNA, and a mutation is just a change that has occurred somewhere in the DNA sequence. So there are different types of DNA mutations, and I know you have to read it on your own, but I want to review them with you in your introduction. So if we come up here, here's the introduction part on mutations. So there's 2 groups of mutations. They can be a frame shift or a substitution. So a frame shift happens when a nucleotide in DNA is either deleted or inserted. So this changes the whole codon reading frame in mRNA, causing that frame shift, and completely changes the amino acid sequence. So a frame shift can be a frame shift deletion or a frame shift insertion. The other type is a substitution. So now, instead of deleting or adding a nucleotide is changed in the DNA. Okay, so it can be changed or substituted for another different nucleotide. If the DNA substitution or the changes the original amino acid that it was supposed to code for, that is called a substitution missense mutation. Okay, if the substitution nucleotide does not change the amino acid from the original one, it's called substitution silent. And if the substitution nucleotide changes the amino acid to an early stop codon from the original, it's called substitution nonsense. Okay, so recap, there's 5 different mutations you can pick from: frame shift insertion, frame shift deletion, substitution missense, substitution silent, and substitution nonsense. Okay, so now that we have that information, we can go on and work on our DNA mutations. So let's go to the work part of it here, okay? So here is the original DNA sequence, okay? This is how it's supposed to normally be. We're going to find, just like before, our mRNA sequence and the amino acid sequence. That is the first step. So here in your Word document, we're going to work on part 3, mutations in the Word document. Here I want to do this part with you. I'm going to do the first mutation with you, and then you're going to be on your own for the rest. So here is our original DNA sequence. So just like before, I like to read it in groups of 3 bases so I can make my mRNA codons. So again, T goes with A goes with U, C goes with G. A goes with U, C goes with G, C goes with G. The next one is TTG, which goes with AAC. Then we have GCG, which goes with CGC. Then we have ACG, which goes with UGC, and then ACT, which goes with UGA. Okay, one more time, let's just use that genetic code. Okay, so I'm going to come back to my lab, this genetic code, and we're going to find the amino acids. So the first one is AUG. Okay, so AUG is methionine, MET. So MET is my first amino acid. UGG. Okay, let's go back. UGG is tryptophan, trip. So that's my next amino acid. AAC. AAC is asparagine, which is esin. So we're going to come back, okay, and that's our next amino acid. CGC is arginine, arg. That's our next amino acid. UGC UGC is cysteine, cis. Okay, so here we go. UGA is a stop codon. Okay, so that is my polypeptide chain, my string of amino acids that will make my protein. Okay, so this is the original. This is normal. This is how this DNA is supposed to function in the cell. Okay, but now something happened to this DNA strand. Okay, and I'm going to show you different scenarios that can happen to the same original DNA sequence. So here's the first scenario, the first mutation that could occur in this original DNA strand. Okay, so if we go back to your lab, the DNA strand is shown here, the mutated DNA strand, okay? And as you can see, the DNA mutation always happens in the DNA strand. So what I really want you to do is compare the mutated DNA sequence with the original DNA sequence, okay? So let's look at it really quick. So T is the same, A is the same, C, A. Oh, here is something different. So we can see here the next nucleotide was supposed to be C, and here it changed to a T. And if we keep looking forward, everything else is the same. So right away we know this is a substitution. That's when we have a nucleotide change. The C got changed into a T. So this is where the DNA mutation occurred. And since there's a DNA mutation, we're going to get a different mRNA sequence, and we're going to see if it's going to change the amino acid sequence. So let's go back, okay? So what I simply did, what you will do in your lab manual and then write it out here in your Word document, is I wrote out the mRNA sequence, okay? When I wrote it out, I looked what changed from the original mRNA sequence. So here, UGG is now UAG, okay? So I know this A was a result of that DNA mutation. So now I have a totally different codon. So AUG is the same, so I know that codes for MET. And before it was UGG, and now it's UAG. So now I have to come back to my genetic code and see what UAG codes for. A stop codon, okay? So I'm going to come and I'm going to write stop for my next codon. Once you reach a stop codon, translation stops, okay? So I would not write anything else for the remaining codons. So that is it. So this is my protein. It just has one amino acid, MET, okay? And now I'm going to compare my amino acid sequence with my original. So my original had 5 different amino acids. Here it only has MET. So is this going to be the same protein, a protein with 5 amino acids versus a protein with 1? They're going to be totally different. So will the protein be affected? Yes, okay? So the only time the protein's not affected if it has the same amino acid sequence. Whenever the amino acid sequence is anything different, the protein will always be affected. Okay, so we already went over that this was a substitution, and when it codes for a stop amino acid, this is substitution nonsense. Okay, so I just did the first mutation with you. Your job is to do mutations number 2 through 5 on your own. A hint I can give you is over here in your lab manual, I wrote out the original DNA sequence and all the mutated DNA sequences you have to work through, okay? They're the same sequences that are all in these boxes, okay? I just kind of stack them on top of each other. This is to help you compare your DNA mutated sequences with the original DNA strand. So remember we did the first one and we saw right here that the C changed to a T. So we knew right away that this was substitution, and then we had to figure out the amino acids to figure out it was a substitution nonsense. Here in sequence number 2, okay, when I'm looking through it, I see that the A looks like it changed into a G. But be careful, there's a big hint, and what I see right away is that there is an extra nucleotide at the end. So see how it stops here? And now I get this extra one. So that kind of gives me a hint that this is an insertion. So when I look at the A to the G, I actually see that the G was inserted, and see how the A got moved after. So ACC is after the G. So the G is actually an insertion. So I gave you a hint for number 2. Number 3 is the same length, so something else must have happened in this mutation. Okay, so I'm looking through, I'm scanning to see where the change is, and here I see the G changed into an A, which we know is a substitution. Okay. Sequence number 4 is the same length, so something had to happen. Scanning through, I'm just comparing this with my original DNA strand. And right here I see the G changed into a T. Okay, so that is a substitution. And then the last strand, if I compare it with my original, it looks like it is a base short. So right away, that is telling me that it's a deletion, and it's a tricky deletion. So we have to figure out where this happened. So again, just scan through, I'm comparing it, right? T is the same, A is the same, C is the same, ACC is the same, TTG is the same, and now here, GGA. So something happened right around here where all these G's are. Okay, so if I look here, the 2 T's are the same, the 2 G's are the same, and the next should be a C. And we are not seeing that C, but we see the GAC after. So it's really the C got deleted, okay? So those are my really big hints. I'm really trying to help you out because I know you're doing this on your own. So I just kind of gave you the hints of what the mutations are. You still have to work through to make the mRNA and the amino acid sequence to see if the protein's going to be affected, okay? So again, I want you to work through all those mutations in your Word document. I'm going to be looking for them. Okay, so here's all your mutations. And then the last are your post-lab questions. So your post-lab questions are very straightforward, and you can just answer them simply in your Word document. You have 8 different ones. 1 through 7 can be answered just straight out. And then number 8 has to do with sickle cell anemia. Okay, so let's just look at question number 8. So question number 8, you're going to read about sickle cell anemia. You're going to see the normal DNA strand for a protein called hemoglobin, okay? So here's the normal DNA strand. You're going to do the mRNA and the amino acid sequence. Here is in sickle cell, there is a mutation. I'm not going to help you with this mutation. I really want you to compare this strand with the original. See where the difference is, locate it, okay? Locate how it makes your mRNA different and how it makes your amino acid sequence different, okay? So again, write these out in your Word document. You're going to tell me which amino acid is different in sickle cell versus normal, and you're going to tell me what type of mutation occurred in sickle cell anemia. So again, just write out your normal mRNA, your normal amino acid sequence, and then the sickle cell or the mutated mRNA and the sickle cell amino acid sequence. Tell me what amino acid sequence is, or what amino acid is different in sickle cell versus normal, and what type of mutation did you observe that happened from the normal DNA to the sickle cell DNA. Make sure you save this Word document to your computer and you upload it to the submission link in Blackboard. This is the end of the online instructions for lab number 7.